Dear student, in this video we discuss about the gene bank. This point belongs to unit number 2nd, recombinant DNA technology. This point is a very new to you, therefore I prepare the video of gene bank. Gene bank are a type of biodispository which preserve genetic material. A collection of seed plants, tissue culture, etc. from potentially useful species, especially species containing genes of significance to the breeding of crops. In plants, this could be done by freezing cuts from the plants or stocking the seeds. With corals, fragments are taken which are stored in water tank under controlled condition. Plant genetic material in a gene bank is preserved at minus 196 degrees Celsius in a liquid nitrogen as mature seed, dry or tissue which belongs to meristems. In plants, it is possible to unfreeze the material and propagate it. However, in animals, a living female is required for artificial insemination. The database of the largest gene bank in the world can be queried via a common website, and name of that website is GeneSAS. Gene banks exist to conserve the genetic diversity of a wild and domesticated organism that human depends on for food, fiber, medicine as well as energy. Over 7.2 million plant germplasm accessions are housed in about 1750 national and international gene banks. In an effort to conserve agricultural biodiversity, gene banks are used to store and conserve the genetic resources of major crop plants and their crop wild relatives. There are many gene banks all over the world, with the Salvard Global Seed Vault being probably the most famous one. Accession is the common term given to an individual sample in a gene bank, such as a distinct species or variety. Most accessions are poorly characterized. Few are ever used. Gene banks manage both genetic resource and information about those resources. Diverse genetic resources are essential to improving the productivity, nutritional quality, and sustainability of agricultural system. Rapid scientific or technological development have changed the way scientific explore and understand natural variation. Gene bank managers must stay abreast of scientific developments to fulfill their responsibility to the public. In an effort to conserve agricultural biodiversity, gene banks are used to store and conserve the plant genetic resources of major crop plants and their crop wild relatives. There are many genes bank all over the world, with the Salvard Global Seed Vault being probably the most famous one. Means all of you know the library. In library, we store the books. Valuable books, these are stored in the books, and when we want that book, we taken from the library by noting the accession number, and we read that book. After reading, it is get back to the library. Similar fashion in a bank also, money bank, we keep our money. Whenever we want that money, we take from that bank. Such type of a same thing is done in this gene bank, but in that the depository is a different one. Depository is present in the form of the gene as well as the material from which the further multiplication of that plant species is done, and in the national as well as international level, the agricultural crop biodiversity along with their wild varieties, wild relatives, their deposition, their conservation is done in this gene bank. Correct? Regarding to history, there are number of scientists who discover as well as who play important role in this gene bank. In that, Sir Otto Frankel coined the term genetic resource in 1968 to aware the plant breeders of this gradual loss of a germplasm. In 1970, in USA, initiation action to collect, conserve, evaluate and utilize the plant germplasm resources when southern corn leaf blight was outbroke. Alphonse D. Kendall was the first botanist to attempt to locate the origin of crop plants. In 1882, he published a book, Origin of Cultivated Plants. In 1926, Nikolai Ivanovich Vavilov, Russian explorer, genetics, agronomist, organized worldwide exploration for collecting the seeds and propagating material of large number of cultivated crops, wild and related species. Then in 1951, in them the scientists proposed the 8 center of origin and 3 subcenter of a different plant species, and the name of that scientist is nothing but Vavilov. In 1961, FAO organized the first international technical meeting on plant exploration and introduction. In 1968, the Crop Ecology and Genetic Resources Unit of FAO was established, while in 1974, International Board of Plant Genetic Resources IBPGR was established in Rome. In 1992, IBPGR was transferred to a new autonomous organization, International Plant Genetic Resource Institute IPGRI, to assist the countries, and that countries are developing countries. While in 1905, conservation of PGRs was initiated in India. 1976, NBPGR was established in New Delhi for conservation of various crop species. In 1985, NBAGR was established in a kernel for conservation of various animal species. There are number of types of gene banks, which is labeled as a seed bank, tissue bank, cryo bank, pollen bank, field gene bank, sperm bank as well as a OHA bank. But we only related to seed bank, tissue bank, cryo bank, pollen bank, field gene bank. We first study the seed bank. The seed bank preserves dried seeds by storing them at a very low temperature. Spores and pteridophytes are conserved in seed banks, but other seedless plants such as tuber crops cannot be preserved in this way. The largest seed bank in the world is the Millennium Seed Bank, housed at the Wellcome Trust Millennium Building in London. Second, tissue bank. In this technique, birds, protocoms and meristematic cells are conserved through the particular light and temperature arrangements in a nutrient medium. This is used to preserve seedless plants and plants which reproduce asexually. Third branch is nothing but a cryo bank. In this technique, a seed or embryo is preserved at very low temperature. It is usually preserved in a liquid nitrogen having minus 196 degrees centigrade temperature. This is helpful for the conservation of species facing extinction. Then pollen bank, this is a method in which pollen grains are stored. We can make plants which are facing extinction in the present world using this technique. By this technique, we can make plants with one set chromosomes. Next one is a field bank, as field gene bank. This is a method of planting plants for the conservation of genes. For this purpose, we construct ecosystem artificially. Through this method, one can compare the different among plants of different species in details. It needs more land, adequate soil, weather, etc. Germplasm of important crops are conserved through this method. For about 42,000 varieties of rice are conserved in the Central Rice Research Institute in Orissa. Along with them, one of the important gene bank is nothing but a botanical garden. Botanical garden over we found that 2500 in 148 countries with about 6.13 million accessions. These are present in a different botanical garden over the world. What is the purpose of this gene bank? First purpose is nothing but a physical facilities for maintaining collection of live plant material, domesticated, cultivated plants, wild plant species, as well as a species which is useful for food and agriculture, as well as which is having other end uses. Their cultivation is important. Then entire plant, seeds, pollen, embryos, meristem, cells or DNA depending on that biology of that species. It is a facility for maintaining crop diversity. Usually this diversity is the form of seeds stored and conserved in a frozen state. Some gene banks use normal household freezer for this purpose. The ideal temperature is between -10 and -20. Each different type is stored in its own container, such as a bottle, a can or a sealed aluminum foil package. Maintaining material in gene bank is often termed ex situ conservation. The definition of ex situ conservation is in this manner: the conservation of component of biodiversity outside their natural habitat. That is nothing but a term as a ex situ conservation. And therefore, example of ex situ conservation, it is nothing but a gene bank as well as a botanical garden. Always a objective type of a question is asked, which is the ex situ conservation method, and below you have to given the number of a technique. You remember well, ex situ conservation is nothing but a gene bank or botanical garden, whereas in situ conservation is the maintenance of viable population in their natural surroundings, a dynamic system which allows the biological resources to evolve and change over time through natural selection process. Both concepts are therefore fundamentally different but are complementary. Gene bank therefore the managed so as to gene bank maintain the genetic integrity of its accessions, make the accessions easily available to users of germplasm, provide the raw material for plant breeding and basic biological research. Accessions of crop wild relatives are particularly valuable as a source of gene provide germplasm for restoration of lost crops after natural or man-made catastrophes. There are various activities coming in that gene bank. The main activities in the development and management of a gene bank include collection and acquisition, then assembling the collection, processing, assessing the quality, viability, health of sample and preparation for storage. In the storage, cold storage, laboratory or in the field. Regeneration and multiplication in that periodically regeneration waiting the increasing the material. Again after that the increasing the material, their characterization and evaluation is done. Then the documentation is done. Inventory, that is nothing but maintaining and making available detailed records, is essential for each and every sample. Then afterward the distribution is done. Clean disease free seeds or other planting material to requistors. These are supply for the multiplication. Means these number of activities is done in the gene bank. Student, I think you will understand the gene bank, the meaning, their activities, where it the purpose of formation of a gene bank. Thank you. We will meet in another video.